Now we're going to take a look at the polysaccharides. Cellulose is the first one we're going to look at. And just to reiterate or remind you from the first video that polysaccharides contain 3 or more monosaccharides linked together. Cellulose is a linear polysaccharide, so unbranched, so there's nothing branching off, and it's made up of repeating glucose units, as are some other types of polysaccharides, and we'll talk about those. But the units in cellulose are linked by alpha 1,4 beta, to do black there, glycosidic linkages. Okay? So cellulose is a part of what make up the cell walls in plants. It gives them the support and rigidity needed to stand erect, basically. And we are not able to digest cellulose because we do not have the enzyme that hydrolyzes that beta linkage found in the cellulose molecules. So it's basically the insoluble fiber in our diets, and that is important to the health of our digestive system. Sometimes people take extra fiber when they have a little trouble here. But as you can see, we've got those beta 1,4 linkages that are shown in blue, which makes it inappropriate for us to, or we can't digest because we lack that enzyme. These are moving slowly here. Don't move. Okay. Darn it. I don't know why this is moving slow, but I just passed a slide that I didn't get to talk about, so I'm going to talk about it here. So the prior slide is taking a look at starch. We are looking at the second form here, or the second type. The one on the previous slide is amylose, which is joined by alpha glycosidic linkages. Both of these starches are. Amylose is found in corn, rice, wheat, and potatoes, and it is a major type of starch. It is linear, and of course we have those alpha linkages. The difference between amylose and amylopectin that is shown here is there is branching. So whereas amylose was a completely linear polysaccharides, we have some branching coming off of the main chain here. But the linkages between those glucose units, and again they are glucose, are alpha 1,4. So we are able to digest these starches, both of these starches. All right, let's move it a little better. I might be doing too many things at one time. Amylose, then, to summarize, is an unbranched or linear polysaccharide that's linked by the alpha 1,4 glycosidic linkages, whereas amylopectin is a branched polysaccharide. The branching linkage is at a 1,6 alpha glycosidic linkage. And we'll take a closer look at that, I think, in glycogen. Both of these molecules can be broken down by the enzyme amylase. Glycogen is the major form of polysaccharide storage in animals, including us, of course. It is similar in a structure to amylopectin, however, it is more highly branched, more branching than in the amylopectin polysaccharide, primarily stored in the liver and muscles, but primarily liver. And when glucose is needed for energy, the glucose units are then hydrolyzed from the ends of the glycogen molecule. Because glycogen is highly branched, there are many ends available for hydrolysis. And this is an example of what a glycogen molecule might look like. Here we have, let's say that's the chain, and we have a branch here, and we have a branch here. So this is a highly branched storage molecule that can be hydrolyzed quickly in times when we need extra burst of energy, like if we're exercising strenuously or we have that fight or flight response and we need a burst of energy, then we can utilize the glucose units in a glycogen molecule. So here's a little interest or some interesting aspect of these carbohydrates. They contribute to what kind of blood type that we have. So there are 4 blood types, AB, 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 and O. And blood type is then based on 3 or 4 monosaccharides that are attached to the membrane of the red blood cells. And each blood type has the monosaccharides that are listed here: D-galactose, L-fructose, and N-acetyl D-glucosamine. Those are all present. However, the 4th monosaccharide in type A blood is N-acetyl D-galactosamine, so that distinguishes it from the other blood types. Type B contains an additional D-galactose unit, so 2 of those rather than just 1. Type AB has both type A and type B carbohydrates, so it's combined the AB type. And then the blood, we all know this, if you've ever had to have a surgery, or maybe a relative needs blood for some reason, you always have to have your blood typed and cross-matched, because the blood of one individual may contain antibodies to another blood type. So those with type O blood are called universal donors because people with other blood types have no antibodies to the type O blood. So those with type AB blood are universal recipients because their blood contains no antibodies to AB or O. Kind of an interesting use of those carbohydrates. And I guess there's nothing on that slide. So I think the next topic is fatty acids.